And then Kersti would introduce.
Hello to everybody. Today's speaker, Małgorzata Słapińska, is a PhD student from Poland, from Warsaw University of Life Sciences. And she got the scholarship from PROM program to join our work group for 2 weeks. But due to Corona situation, this visit was postponed until we will see, until so far. But however, she will give a presentation today about part of her research. So, the floor is yours. Thanks.
Okay, thank you very much for the introduction. As Kersti said, I am a PhD student from Poland. I'm from Warsaw University of Life Sciences, and I work in the Department of Remote Sensing and Environmental Assessment. So today I will speak about modeling of phytoplankton and water quality in Biebrza River Oxbow lakes in northeastern Poland. The aim of this study and of this project, because it's a project I wrote for my PhD studies, is to retrieve empirical formulas for phytoplankton groups and water quality for oxbow lakes in the lower Biebrza basin with the use of HySpex airborne imaging spectrometer camera. Okay, so first I want to introduce you about the area of the research and the oxbow lakes I work on. There are several types of oxbow lakes. The most general classification is by the connection with the river. So we have got oxbow lakes which are connected with both arms with the river, or with one arm. With one arm, it is called semi-lotic oxbow lake, and here we have got an example. And sometimes there are oxbow lakes which are connected with both arms, and they are called lotic oxbow lakes. And here, this one, oxbow lake, you will see it after with formula I used on it. It's oxbow lake which is not connected at all, and it is called blended oxbow lake. So here we have got a photo from semi-lotic oxbow lake in Biebrza, from the study site. There is a lot of water there, and it is in quite good condition. And here we have blended oxbow lake. There is much less water in it, and it is more turbid, and the quality of this water is worse. So the area of the research, it's a river in Biebrza National Park. Here we have on the map. It is the biggest national park in Poland. It was established in 1993, and it is located in the northeastern part of the country, situated along the Biebrza River. This is the biggest wetland in Europe. This is one of the biggest wetlands in Europe. It is very low contaminated. The water is clear. The river is naturally meandering, and this area is used as a reference site for many research, for example, for the Netherlands or other Western European countries. In 1995, the park was also designated as a wetland site under protection of Ramsar Convention. And it is a place where a lot of different species of birds under protection, and it is also used for recreation. And this is the lower, this is the Biebrza Park border, and Biebrza is, there are three parts of Biebrza River: the lower basin, the middle basin, and the upper basin. And my research was placed in the lower basin, which is the most natural one, and where the Biebrza Park is located. So I use this area, and the dots are the points where which was measured, and from where the water was taken. So first I want to talk about the methodology. Sorry. About the methodology of the research. On 1st and 2nd of July 2015, the HySpex data was acquired by the company we work with, and it was atmospherically collected by Atco model, and there the reflectance data was acquired. A few days later, the field campaign was conducted. The such disk depth was measured together with electroconductivity and pH, and water samples for further analysis were taken. And in the lab, the phytoplankton was analyzed together with BOD. And from that measurement, the statistic formulas were acquired in R, and from the formulas, the maps were so for the analysis in R, 166 channels with the wavelengths between approximately 400 nanometers to 960, and this round was used because of the stored information about water within this round. And for each channel, the combination of for each channel from 166 bands, any combination was made, and the normalized difference index for these bands was counted with the formula similar to NDVI. So further in the presentation, I used the NDVI formula. I recall to this formula. And another stage was correlating each normalized difference combination with the result of such disk depth, electroconductivity, and BOD, and phytoplankton groups measured in situ. And after that, formulas were in the form of linear regression or multiple regression. And also in those formulas, I used the normalized differences index to make more those formulas more universal. And during the field campaign, the water samples were taken from the surface of the water, which is not typical for phytoplankton analysis. And to determine the taxonomic composition of phytoplankton, the individual were counted in tubular plankton chambers using an inverted microscope. And their observed organisms were classified to the groups. Here is the vision from the microscope. My friend counted this for me. And here are the formulas for water quality. For the certificate that I use two wavelengths with R for 0.78. For electric conductivity, I use these wavelengths with R square 0.53. And for biological oxygen demand, I use those wavelengths with R square 0.80. And here are the graphs for such as depth, electric conductivity, and biological oxygen demand. Here is the parameter, and here is the correlation of NDVI of two bands. Here I've got a map for such as depth. This is the Atlantic Oxbow Lake I mentioned before, and this Oxbow Lake it's connected with the river with just one arm, but its quality is much better than this Oxbow Lake. And here I've got BOD for both river. You can see that the water in river is clear, and in the Oxbow Lake it's becoming more turbid. And here in this Atlantic Oxbow Lake, which has the worst water quality result from all of the study area, you can see that water is has higher values and its water is less pure. Here I've got results for the Cyanophyta phytoplankton group, which is in English blue-green algae. I use different wavelengths for the formula. The formula is for the band 524 nanometers, 540, 470, and 579, which are located here in the spectrum. The R squared was 0.61, and the F statistic and P value. Sorry, can you hear me now?
Yes, now I can again hear.
Okay, I think there is some problem with the connection. Maybe I'll go with lower quality of the video. Okay, so here on the graph with the blue, you can see the area with the best tolerating values. the more intense the color, the higher the correlation. So for the analyze, I use wavelengths from this area. And for the chlorophyta, I also use different for different bands. 636, 649, 659, and 713 with R squared 0.91. And the graph illustrates the different wavelengths which were correlating. So the idea, the way we made these formulas, we wanted to have some bands from this area, but also from this. So the formula will be more universal. The bacillary phyta, the atoms, have a similar graph and similar bands correlating like the chlorophyta, but here we use more bands. We used 6 different bands. Also 425 nanometers and 873 nanometers was used, and the R squared was 0.85. Euglenophyta, euglenoids, have these bands correlating, and the number was 512, 694, and also 520, 690, 560, and 704. Our squared was smaller, it was 0.48. And the last one, which was measured, it was also multiple regression with bands 470, 580, 460, 560, 470, and 620, with R squared 0.49. So this, with the previous one, has the lowest correlation from the phytoplankton. And the result was applying those formulas on the map from HySpex, on the reference map of the HySpex camera. And here, similar to the previous map for the SHD step, you can see that the river has better quality, maybe not better quality, but lower algae concentration where the water is flowing. Here, in the places where river is meandering, the color is changing, so the water is standing there, it's not flowing, so it's a better place for the phytoplankton groups to develop. The derived formulas have acceptable high R square, which is depth 0.98, electroconductivity 0.53, DOD 0.80, and phytoplankton from 0.91 to the lowest 0.48. And most phytoplankton groups were correlated with different wavelengths, and this is due to the fact that each phytoplankton has different pigments, and therefore they have different spectral characteristics. So they can be monitored by the formulas, maybe not like the different species, but to the general good. And chlorophyta and bacteriophyta were correlated. The similar wavelengths were used, and in the formulas to make them distinguish, I used 4 different wavelengths for chlorophyta and 6 different wavelengths for bacteriophyta. The dominated phytoplankton group in most cases, in most points where the water is taken, were chlorophyta. This is because it was the beginning of June and high insolation. It was around 36 degrees. There's also a lot of albanophyta, and this is due to organic pollution, especially nitrogen. And the distribution maps confirmed characteristic entropic state of the different types of oxbow rake. The elotic and semi-elotic oxbow rakes usually are more trophic than elotic oxbow rakes. And this was the research I conducted 2 years ago. It was a part of my PhD, and I wrote the small internal grant to make it. And now with my team, we are preparing for a new project, which will take place in Zalew Koronowski. We just got the grant acceptance, and the project will start in 2021. This is a research and development project with commercial company, and we use drone and hyperspectral camera with LIDAR to monitor Zalew Koronowski, which is it is all one water body. Here we can sail. It is connected here, and the river quality is very diverse in this area. And due to this shape, it is not possible to monitor it with geostatistical methods. So remote sensing, it's a good idea for this lake. And in this project, we want to monitor both water quality and forest condition. We have 2 teams, one for water and one for forest. And the aim is to build a system with drone and cameras to monitor threats to the environment. For water, it is cyanophyta, blue algae, and for forest, it's bark beetle. And as I mentioned before, we will start it in 2021. So if you have got any suggestions about this project, we are open for them. And this is all. And thank you very much for your attention.
Thank you.
Thank you.
Now I will stop recording.